So the first question, and I think it's most relevant to Sophie. So if we could bring Sophie in. Sophie, you're at the forefront of delivering cell therapies. What do you see as the trajectory for cell therapies, or the future for cell therapies, given that at this stage within the UK and the EU, we've only really got 2 that have been approved for patients?
Well, I mean, I think the future's now, as you've said. We've got 2 approved therapies, well, more than 2, because there are a couple of a number of CD19 CAR constructs, and now the BCMA CARs are coming through, which is really exciting. If the question is, are we going to be giving cell therapy for solid tumor oncology? Yes, 100%. Will it be genetically modified cells? That I still don't know, but we will be delivering TIL, tumor infiltrating lymphocyte therapy, which is procurement of tissue for isolation of infiltrating lymphocytes, expansion, and then delivery. And this is complex therapy because the patients require a lot of chemotherapy to get them ready for treatment. And FDA licenses for melanoma and probably cervical cancer are expected this year. And the projection from NHS England is that we'll be looking at a similar approvals in the UK in the next sort of 2 years, I would say. And there are plenty of other advanced therapies that sit on NHS England's watch list with respects to commissioning in the next few years across the most fantastic spectrum of different indications. And I absolutely think that cellular therapies will form a significant part of what happens and what we're doing clinically over the remainder of my career.
Fantastic. Yes, it is. I mean, both of us are obviously privy to what we see potentially coming through NICE, and it's exponentially growing, which is great for the patients that very much need it. I think that leads on to a question that I have for Jonathan. And from an industry perspective, Jonathan, what do you see as the key challenges in advancing and accelerating cell and gene therapies through to the bedside?
How long have you got? The key challenges, I think you can break them broadly into 2 buckets. Immunology for viral vector delivery, being able to get around the immunological barriers that we have. And we've seen some incidents emerging in the AAV space, which really show us where the hard stop is around the top doses that we can give. We just can't seem to get enough vectors on board to target these large tissues like muscle. So that's a kind of efficacy perspective. But then from a manufacturing perspective, there are many broad. In the vector space, it's largely about scale, and we're getting better at those sorts of things, but it's still very expensive and protracted in terms of procuring the materials and getting the licenses and freedom to operate space that you need in order to be successful in the space. And in the cell therapy space, of course, largely driven by the autologous supply, which is terribly difficult to scale. And I think over time, we'll see that breaking down the technological barriers, but then shifting across to an iPSC-based type platform, which we see groups like Fate Therapeutics now being successful with. So it depends which area you're in. So immunology, supply for vectors, and switching away from autologous in the cell therapy space. And then there's, of course, the whole regenerative medicine space as well, where I think it's largely about differentiation and cell selection to make sure you have a safe and pure product.
Great. Thanks very much, Jonathan. And a nice segue, in fact, to a question that came up on Slido to Ash. You've just completed the delivery of your monocytes within your phase 1 study. Monocytes manufactured within our GMP, but at a relatively small scale. Or a very small scale. So what do you see from your perspective in the cell therapies and perhaps in particular to your monocytes in scaling up that for wider use and autologous or allogenic approaches going forward?
Thanks, Shaun. So monocytes don't divide. So the cells that you can isolate from the blood or from leukapheresis is what you start off with. Paradoxically, it's actually easier to work with larger numbers of cells with the equipment that we use. So to simply leukapherese a patient and isolate the monocytes, we can quite quickly scale up. But as Sophie mentioned earlier, leukapheresis and that service is something that's needed at GTT. Going further forward, as Jonathan mentioned, iPSCs I think are going to be really interesting. So the ability to generate autologous iPSCs, and then from those cells to then differentiate those into monocytes, for example, and other types of cells, will really allow us to scale up our products. And once you have a well-defined manufacturing process, there are multiple other manufacturing centres around the UK that you can easily transfer your technology to, providing that you've shown that you can do it reproducibly in your own unit.
Great, thanks very much, Ash. Again, a very useful segue to another question that came up that Robin perhaps can address. Robin, we've spent the morning focused very much on cell and gene therapies, but there are occasions where next generation antibodies, next generation recombinant proteins could in effect be considered advanced therapies. But we don't have the capacity or the infrastructure to manufacture within our GMP facility. So what would you recommend to those researchers that are active in that area? Where should they go, or where could they go in order to advance that element of advanced therapies?
So I certainly think that those sort of approaches can also fall into the advanced therapy space. And I think the key issue here for our Advanced Therapies Accelerator is whether investigators are limited in establishing first-in-human trials because of the inability to obtain ATIM starting material. Now, for monoclonal antibodies and recombinant proteins, there are a lot of CDMOs that have the capability and technology to be able to provide that relatively cost effectively. And so I wouldn't propose that we should be fulfilling the service of a CDMO that can be contracted out. So I think the Advanced Therapies Accelerator is for innovation, cutting edge therapies that require the establishment of new leading edge technologies.
Great, thanks very much, Robin. Giovanna, you were one of the first researchers to develop a cell therapy within our BRC GMP unit, which is now obviously co-located with the ATA. How do you see the ATA as a whole, so including all our platforms? How do you see that being important to your research, and in particular your Tregs and potentially other cell types that you're working on and wanting to deliver into early phase trials going forward?
I mean, I think, my experience has been extremely positive already, and we achieve a lot. And as you know, a lot of other regulatory T-cell trial have been submitted. It means, I think the advantage of this Advanced Therapy Accelerator is mostly a sort of improving the network between all the different structured platform that we need. But, I think also is the expansion of a program that has been already extremely successful from the beginning. And Katie and I think, are the only two probably that started together, from the first BRC. And I think it's just the number of these clinical trials that are increasing and very successfully. And, the other things is that what already other speaker mentioned is the stem cell aspect, whether we need at one point a Treg that is off the shelf, where we need an iPS that, can help in rendering this approach also a little less expensive. And I think, again, talking about the other program and the stem cell in particular, it is can be extremely helpful.
So we've run out of time for any further questions, but thanks to our speakers for taking those and giving very, very interesting and informative answers.